So when does this go from being... Does this seem reasonable so far? Okay.
Yeah, actually, I was thinking, I don't know whether this is the right point, but one thing that I find helpful in thinking about when we talk about the hypothesis is that it typically has two components. We tend to forget about one. So one is the hypothesis we happen to be thinking about.
Right.
The other one is the models that we use to test it.
Right.
Right. So the specific statistical test we used has tons of assumptions associated with it. And all of this you could read with that additional conditional on it, essentially, all of those statements, given the modeling assumptions I made, and given the hypothesis I had, and given the alternative hypotheses I considered.
That is a very
people trying to get the same data.
That is a very, very good point. I mean, that's a very, if you decide, if this isn't putting you off and you decide to plunge more into that, that's exactly the kind of thing you'll have to think more about because, and in fact, I mean, this is the kind of thing you have to think about in research. You can't really, on the whole, get away from not only testing a hypothesis, but testing a hypothesis in a particular way, using some kind of framework, some kind of model. And it's easy to forget that you're doing that, but it's important not to forget that you're doing that. So I think that's what you're saying, right? And that's a very, very important point. So, okay, so let's try and make this a little bit less kind of weirdly abstract and a little bit more concrete and relate it a little bit back more to fMRI. Okay, I'm sure there must be some questions and confusions and puzzlement at this point. It's entirely... If you're feeling any puzzlement at this point, that just shows that you have a normally working human brain. And I'm not kidding, because the human brain is really... There's a lot of studies actually showing this. The human brain is really not well set up for thinking about this kind of thing. It just isn't. Okay, so do not be alarmed if you're finding this confusing. Okay. Okay, so here's an example. This is the kind of thing that you will definitely get 1,000 times in medical school, but it's actually important to think about in general, and it crops up in real life too, I would say. It certainly crops up in science. So these two things, probability of hypothesis given data and probability of data given hypothesis, I can see that they're written the opposite ways around from each other on the on the projector, but what does that really concretely mean? When would these two things come apart a little bit? Well, they especially come apart when you do lots and lots of tests for something that doesn't happen very often, okay? And guess what happens? Guess what fMRI is, in large part, okay? It's running lots and lots of tests for something that may not be happening very often. In this case, activation. But a more concrete example, which is often used, is, say, testing for a rare disease. Actually, this really does crop up in everyday life, because just recently, there's been a whole bunch of news stories about, the American Medical Association no longer recommends certain types of screening for cancer. No longer recommends that, I forget exactly what it is. It no longer recommends that everyone should get, men should get screened for prostate cancer every year, and women should get screened for mammograms every year or something. It's meant to be every 5 years or something. I forget what it is. Please look up what it is and don't take it. But people go nuts when these recommendations get made, because they say, well, no, this is how dare you take away my screening from me, right? I need screening. I don't want to die of cancer, please, thank you very much. I want to get screened every year. And the medical associations have a really, really, really difficult time explaining, and I don't even know if they've I haven't really looked into this, but I don't think I've ever even seen a really concerted attempt to try and explain it. These are the kind of reasons why it's not necessarily a good idea to just screen millions of people every year for a disease that some only relatively small proportion of them will have, and that if anybody does get a false positive saying, oh, we think you might have cancer, they're going to literally have bits chopped out of them, and it's kind of a big deal. So it's very, very difficult to explain that. So this is not just an abstract question. But let's make it a little bit more abstract, because probably cancer is something we do have to worry about. We don't have to worry too much about getting bubonic plague. So let's talk about bubonic plague and not feel uncomfortable. Okay. So so suppose that 0.1% of the population has bubonic plague. I mean, luckily it's probably a lot less than that. Okay. And suppose we have a really, really, really good test for plague, which has 99% specificity, meaning that it has the false positive rate is 1%. Okay? And that's about as good a test as you'll ever get. You're not going to get anything much better than that. Okay? So, and then the classic question, and seriously, any of you who are interested in going to medical school, learn this, because you're going to get asked this 1,000 times. And you'll only get it, some of your answers will be wrong. Okay, so suppose I test 1,000 people. And to show that this is relevant to fMRI, just cross out the word people and put voxels, okay, and cross out the word plague and put activation. Okay, okay, so if I've got 1,000 people, 0.1% of them, this is called the base rate, how many of them have it. We have a low base rate here. 0.1% of them has a plague, okay, so one of those people is going to have plague. There's actually some work by a guy called Gigerenzer showing that when you put things in terms of probabilities and percentages, it's extremely non-intuitive. But when you put it in terms of concrete counts of people, it becomes more intuitive. So I'm hoping that he's right. Okay. So and we test them all, and we've got 99% specificity, meaning we've got a 1% false positive rate. So 1% of all these people who we test are going to falsely come up as showing that they have bubonic plague. So that's going to be 10 people. Okay. So so altogether, okay, I've got 10. I suppose altogether this is going to give 11 people, actually, not. Okay, I didn't add this up, right? Okay, yeah. But, sorry. Okay, so 11 people come to me saying that we have plague, okay? Oh no, I got plague, I got plague, I got plague. And only one of them actually has the plague, right? So what looked like a really, really good test, sorry, I got these numbers wrong. Look, I'm trying to teach you stuff, and I got the numbers wrong, okay. So what looks like a really, really good test, and in fact is a really good test, you're doing very, very well if you have a test that has 99% specificity. It's kind of hard to get a test that good. Okay? If you apply it lots and lots of times to something that's quite rare, you're going to just be swamped with false positives. You're going to get way more false positives than actual real hits. But try explaining that to somebody who's just tested positive for bubonic plague, right? Say oh, yeah, we got your results back. You tested positive for bubonic plague, but you really don't need to worry. We really don't recommend that you do anything rash at this point. We're going to monitor you a little bit and see how you're doing. Okay, they're going to be no, give me all of your antibiotics, give me everything you possibly can have, right? This is the problem with screening all the time for cancer earlier. Okay, this is the problem that afflicts fMRI or any type of study in which you're measuring a whole bunch of stuff, you don't necessarily know in advance where to look, and you're doing lots of tests. So here, we're screening a whole bunch of people. We don't know in advance who has the plague, but we have to deal with the results that we get. So a similar kind of thing happens in fMRI. It's worth pointing out that all of this is only the case if your ultimate thing that you ultimately really care about is, is a voxel active or is it not active? And we've spent time in some of the previous slides saying, that's a step along the way. It's not maybe the ultimate thing we care about. It might be more interesting to look at patterns, which is something we'll be talking about a lot. We already have talked about somewhat. But even still, it's definitely a step along the way. So unless you have a good way of actually figuring out, what these actual patterns are, which things are active and which things are not, you're kind of in trouble. So it's important to think about this. So this is the problem of multiple comparisons. So cross out the word people, replace it with voxels, cross out the word plague, replace it with activation. This can happen in fMRI very easily, and I'll show an example of that in a minute. Okay, so I'm only now. This is maybe partly because I was told that I was going a little bit too easy in the last one, but I'm now just going to try and mess with your heads just one more time. Okay, which is. Don't worry, by the way, there's not actually, you don't have to have an answer to this because nobody has an answer. Oh, I just gave away answers. Okay, is there a single widely agreed upon answer to the question? Would anyone like to say, and by the way, do not worry because I'll tell you that there is no widely agreed answer to this, which is actually kind of shocking if you think about it. Would anyone like to say, and don't worry if you don't want to, because I've kind of set it up to make it sound impossible now. If I say I just looked at my weather app on my phone. It says 10% chance of rain tomorrow. What does that mean? What does it actually mean to say there's a 10% chance of rain tomorrow? Now this is actually kind of a philosophical question. I know that at least one of you is into philosophy, probably more, okay? But this is actually a really, really, really difficult question, which, if you think about it, is actually a little bit shocking. This is again not a reason, to say, wow, no one really understands probability, or at least nobody can agree upon answers to probability, or the best way of doing statistical testing, so let's chuck it all out the window. I'm not saying that at all. I'm saying that these are kind of difficult and somewhat murky areas, and the real criterion of whether you found something real is can other people replicate it. All of the statistics and all of the probability is really just kind of a means towards trying to get to that end. Just like putting on a seatbelt is a way of trying to make it a little more likely you're going to get home safely, but it doesn't really guarantee anything, and it's not really, it's not where the absolute core of the issue is. So anyway, so this is actually really a true statement. If any of you ever want to read about this, there's a whole entire libraries full of books about philosophy of probability and interpretation of probability. That if you, and this is especially problematic if you say the probability of single events, like will it rain tomorrow? Not like, how often will it rain in Rochester in the month of June in the 20th century or something like that. Okay, there's no widely agreed upon answer of what that actually means. Yeah, so this is kind of difficult. So if you find, but the good news, so that's the bad news. Okay, the good news is that although nobody necessarily has a widely agreed upon answer of exactly how to interpret these things called probabilities, you can actually use them just fine, even if you don't know exactly how to interpret them. A little bit like, which sounds strange, but is actually true. A little bit like, I don't want to get too philosophical here, but if I say, what is a number, right? There's not actually any particularly well agreed upon answer to what a number is either, just, while we're getting into this kind of thing. But does that stop you from adding up, I've got 2 apples here and 3 apples here, how many apples do I have? No, it doesn't. Okay? But if you say, what is, now I have 5 apples, what is the number 5? It's kind of a hard question to answer. Okay, so this is kind of a little bit like that. So the fact that there's not really an agreed upon answer to this doesn't necessarily get in the way, but the fact that there's still disagreement about what the best way to do statistical tests is actually is a very live issue, and people really do. That's not just an abstract issue of kind of philosophical definitions and the meaning of concepts and stuff like that. That's actually, people are really fighting like right now about what the best way to do statistical tests is. Okay, so anyway, but the crucial point is that there's that fMRI suffers from this multiple comparisons problem, so we have to do something about it. So forgetting all this kind of murkiness, is that part reasonably clear? Like similar to the reason why, if you screen 1,000 people for bubonic plague, you're going to get some false positives. Okay, so there's different ways of dealing with that. I'm just going to kind of briefly describe what they are. I think it's more important to, it's actually easier to see in a picture what these are than to describe them, so I'll do that in a minute. But there's a famous study which is kind of infamous, which probably quite a lot of you have heard. Have you heard of this? How many of you have heard of this? Okay. So no disrespect. I know this is going to go on YouTube. Well, I doubt very much that very many people will watch it. But no disrespect to you guys who did this study. I actually know a couple of you. But this study is really annoying because it's here's what they did. Okay, it's kind of funny and everything, but it's a little bit annoying to me. So they took a dead salmon, they put it in the scanner, and they said, hey, let's run a test on whether this salmon is, sensitive to whatever stimuli they were showing. They actually flash things up on the screen, okay, in front of the salmon. And if you don't do any multiple comparisons testing, then you get some false positives, and here they are. Okay, so here's some false positives. And if you do multiple comparisons testing, then those go away. Okay? So the punchline is, hey guys, let's do multiple comparisons testing. Let's correct for multiple comparisons. But everyone knew that anyway. Okay, so that's why it's actually kind of a little bit annoying. So you'll see journalists especially talk about this a lot. I mean, I set a paper by reading by one of these guys. So I mean, it's kind of a service in the sense that it reminds people. It's like saying, hey everyone, remember to put on your seatbelt. Everyone already knows that they're meant to put on the seatbelt, except in New Hampshire, I suppose. But it's not like a discovery to say we have discovered that people should put on their seatbelts. Okay, so but it kind of gets written about as the world of fMRI, I mean, you'll see this journalists write about it, because who can resist writing about a dead salmon, right? I mean, everyone needs a good punchline in their story. Where the world of fMRI was recently reminded of how important it is to do false positives by scanning with dead salmon. Well, everyone knew already. Okay.
Was everybody doing that?
Huh?
Was everybody actually doing these comparisons?
Very good point. Not everyone was doing it, but everybody knew they should be doing it. Okay, so that's a crucial distinction. Thank you, Florian. And it's just like seatbelts. Okay, okay, it's really just like seatbelts. Okay, because everybody knows they ought to wear a seatbelt, but not everybody wears a seatbelt. Okay, so so it is kind of like that. Yeah.
Does it
So.
have a change on? I'm curious, actually.
Well, the bigger change. Okay, so I'm about to get to the famous Voodoo correlations thing. Okay, so which I'll get to in a minute. The Voodoo. To skip ahead a bit, there's a different thing which is much less obvious and is much more of a real problem, and actually has changed people's way of doing a data analysis, which is this thing called Voodoo correlations, which I'll get to in a minute. And that's much more substantive, and that really, was not just saying. That would be like, saying, hey, everyone wearing a seatbelt, if people didn't even know that there were seatbelts. Okay. Okay. So what are different ways of correcting for multiple comparisons? So this, I like this picture. Okay. So just as I just dissed the dead salmon people, but this is but I set one of their papers, and this is a picture from the Bennett et al. Paper. Which actually very nicely illustrates different types of correction for multiple comparison. And this is using this example, I mentioned earlier that you can just, if you really want to see how these things work, a good way to do this is just run simulations, because in real data you can't specify, I'm going to have exactly this much signal and exactly this much noise, but you can if you just simulate it, and then you can see how things behave. So the real Here, because it's simulated, we have the luxury of saying, so pretend we have some two-dimensional slice of brain here with different voxel activations, and there happens to be a perfectly square shape of activation in it, okay? So I'm just going to pre-specify that, here's our signal. The real signal is that there's a little square of more active voxels, 20 by 20 or whatever it is, but everything is covered with random noise. You can probably see at the back that there's this kind of speckle on top of this here. So then you can say, what do different types of multiple comparisons correction do? So why is this a multiple comparisons issue? Because if I'm trying to ask the question, how much activation is there at this voxel? How much activation is there at this voxel? How much activation is there at this voxel? I'm basically just going to kind of say, give me the active voxels. I'm going to take some threshold, literally a statistical threshold, and say, everything that's above that threshold I'm going to say is active. Okay. So in the real truth of the matter, these guys here are the only active ones. But I don't know if you can see, but there's some pretty bright voxels out here that is just in the noise region. Okay. So if I had hit some kind of threshold, probably you probably can't see this very well. There's a little bright bit here, a little bright bit here, a little bright bit here. Those little bright bits are going to come up as above threshold, and those will be false positives. Okay, and so that's what happens here. Okay, so this is like the kind of dead salmon case. I mean, you have to hand it to them that it's a very memorable example, at least. Okay, so if you say, okay, I'm just going to say, give me everything that passes my threshold test of p is less than 0.05, I'm going to get the stuff in the middle, the real signal. Not all of it, because some of the noise actually makes it go down. There's some little holes in here. But I'm going to get lots and lots of things out here too. So here's lots and lots of false positives. Now there's a type of correction. So the most kind of tough-minded, if you like, type of correction you can possibly do is Bonferroni correction, which says instead of looking at a threshold of 0.05, take your threshold value and divide it by the number of tests that you're doing. Okay, so say, I don't know how many dots are on this grid, say it's 100 by 100 or something. Okay, then now I'm doing 10,000 tests. So I say now instead my threshold is going to be not 0.05, but 0.05 divided by 10,000, so 0.0005 or something. Okay, so some super duper duper strict threshold, and the result will be that almost nothing will show up as active. Okay, so you can guarantee you're not going to get any false positives just by saying no to everything. Right? I can produce you the world's most false positive free test for bubonic plague by just having a book that says he doesn't have bubonic plague. Right? Guaranteed no false positives. Not a very useful test. Okay? So Bonferroni correction can sometimes end up a little bit like that. Okay? Now, but that's just you might say, well, tough. You might not like the results, but that's the way to do it. It turns out there's actually a reason why that's not actually a very good way of doing it, and that is because I'm not really running 10,000 tests. Remember the example of like measuring the height of the tide every 2 minutes. You're not really getting a fresh piece of information every 2 minutes. Okay, so this I'm pretty sure they've done it to this, and fMRI data in general is kind of a little bit smooth. So what's going on in one voxel tells you something about what's going on in the neighboring voxel. Just because you're not measuring perfectly just this little spot, you've got a kind of bit of a spread in what you're measuring, and people's heads are moving a bit, and they're all being served by those big blood vessels and stuff. So it's not the case that you have all of these little tiny, completely separate points of MRI signal that you're measuring. Just like if I measure the height of the tide every 2 minutes, I'm not really measuring a new thing every time either, even if I tell myself I am. So instead of dividing, so what is called family-wise error correction. This is built into all the standard imaging packages, including this one SPM we're going to play with on Thursday, although we're not actually going to play with this part of it on Thursday, I don't think. It says, okay, don't divide by 10,000. Divide by the effective number of measurements that you actually made, where the effective number of measurements that you actually made is something that kind of takes into account the fact that there's a little bit of kind of smoothness here. There's a bit of correlation between one point and its neighboring point. And there's all kinds of like fairly fancy formulas. It's called Gaussian random fields, if you're into this kind of stuff, for calculating how many measurements you're actually really making, given how smooth things are, and then you divide by that number instead of dividing by 10,000. So that's called family-wise error correction. But notice what this does. I mean, this is why I'm looking at a picture, I think. So, thank you, Bennett et al., for making this picture. This is, I think, is a good indicator of why this still might not necessarily be what you want. Because intuitively, if you say, okay, if I've got a threshold value of 0.05, I kind of want it to be the case that, if some voxel comes up as active, I can say, yeah, there's only a 5% chance that I'm wrong with that voxel. Okay? And you might think that that's actually what family-wise error correction does. Okay? But it doesn't do that. Okay? This is yet another of the kind of non-intuitive things going on in this area. And I remember I was very surprised when I found out it doesn't do that. I'd always thought that's what it does. Okay, it doesn't do that. It doesn't say for any given voxel, I got a 5% chance of it being a false positive. It says, I have a 5% chance that there's going to be any false positives anywhere at all, which is a different thing. And notice, by the way, here, this area outside the square is the kind of false positive region. There's no false positives at all. It's completely black out here, but it's pretty black in here too. Okay, there's not much activation going in here. So this is also very conservative. This is not as bad as the test for bubonic plague that's just a printed book saying definitely doesn't have bubonic plague. But it's not. But you can see why you're kind of throwing out the baby with the bathwater a bit here too. You're kind of saying, okay, any kind of multiple comparisons correction is essentially saying, hey, turn the threshold up a bit here. Make it tougher. And this is maybe making it too tough. Maybe depends what exactly. There's some times in which you really, really, really don't want to have a false positive. And for instance, if it would lead to someone undergoing surgery for cancer. There's sometimes when you don't really care if it's a false positive. What's an example of that? Credit card company rings you up and says, we think there might be some fraud on your account. That happens to me quite often, but I think there was only for one time. Okay? I'm pretty happy. I don't mind. I'd rather have them call me up and say, we think there's some fraud. Could you verify this purchase? than someone actually do fraudulent purchase on account. So, I'm not going into the operating room because they're doing a false positive here. So how strict you want to be depends on what the question is. Okay. So the thing that I always intuitively thought that family-wise error correction did is actually what this thing called false discovery rate does, which is says, okay, for any particular voxel that happens, I only have a 5% chance, if that's what your threshold is, of it being a false positive. And you can kind of see that here. You're getting a pretty good job of getting most of the activation that is the real signal. We know it's real because it made it that way in this simulation, all these guys did. And you're getting a little bit of false positives around the side, but that's kind of the price you're willing to pay. So this is actually quite a popular technique these days. But again, exactly what you do depends in part upon what question you're asking. And I'd also like to just re-emphasize that this is all, if what you really care about is the patterns and not just whether an individual voxel is active, then this is just a step along the road. Yes?
Yeah, so I was going to ask in the context of fMRI, and maybe I could get to this or maybe not. But you can imagine doing this, right? And then you say, okay, there are some risks, right? There could be some, like you said, we reduce the false positives, but there still could be some, like in the outside. But then you could also do like some further, like for instance, cluster correction. You could say, well, only give me voxels that have neighbors that are also activated similarly, and give me maybe 50. You can use some arbitrary number.
Right. That's a very, very good point. So those of you who, and we'll be doing this later on. Those of you who actually play with real fMRI data, as Frank does, will know that there's actually a couple of different ways you can threshold. So far, I've just been talking about thresholding in terms of how bright does a pixel have to be or voxel have to be before I say yep, it's active. Okay, but very, very valid point that if genuine activity really is smooth, okay, I mean this is a connected region. There's actually a chunk of tissue that's active here. Then you can set your test to say, don't just give me things that exceed some height threshold. I want to get chunks of tissue too. I want to get multiple voxels together, and that's a different type of thresholding, and those two kind of interact with each other in interesting ways. And there's all kinds of
there are also built-in packages that allow you to say for a given contrast, faces versus places, because everyone likes to use that example.
Right.
You can take your whole brain voxel map, and you can say, given this map and sort of simulating these t-values that I get, what is the probability that they cluster together like the map I'm getting here?
Exactly. No, that's a very, very valid point.
So there are some built-in ways that you can also test.
Right. No, that's actually, so I've been, no, that's a very valid point. So I'm kind of, there's a whole extra level of kind of complexity here where you can look at this kind of spatial extent.
Yeah, right.
And that's important to, that's very, actually, thank you, that's very worth bearing in mind too. But it's essentially driven by the same problem, but it's a kind of more sophisticated way of trying to deal with the problem, and there's packages. Actually, that's also built into the packages these days as well. Okay, so that's the problem of false positives and different ways of dealing with it. So there's only just a little bit of time. We're going to just quickly talk about Voodoo correlation. So this actually this was a big splash when it came out a few years ago. I don't know, did any of you used to read like science blogs and stuff back in 2009. Blogs did exist back then, believe me. Anyway, if you did, this was like a big thing. So this guy, Ed Vool and his colleagues, and also simultaneously, I put this reference up later, Kriegerskorte, who's one of the really great researchers in pattern-based fMRI, also came up with this similar problem. He called it double dipping. They said, well, there's a funny thing that's happening in the way people are doing data. And this is different from the dead salmon thing, because this is really a problem that people really did run into, and that people really didn't even realize was a problem. Everyone knew they were meant to wear their seatbelt, even if they didn't. Everyone knew they were meant to do multiple comparisons correction, even if they didn't. People didn't even realize this was a problem. Okay? So ROI stands for region of interest. So you run some test. Okay, so here's a region of interest, right? You say, okay, I found areas that are active. Okay, here's my active area. Okay, good, I found it. Okay, now let's go and look in that area, and let's see, for instance, how it correlates with behavior. Well, the problem is that my criterion for finding this region in the first place was probably, find me an area that correlates well with behavior. Okay, So if you say, well, find me an area that correlates well with behavior. Once we've found it, we're going to go look inside that region. You already know in advance that it's going to correlate with behavior because that's how you found it. And you might say, okay, well, that's fine. We're just kind of taking a closer look at that region. Having found it, now we're going to go and look at a bit more. It's like, I go into a bookstore, I find the book I want. Having found the book, I go and open the pages. Okay, I'm just looking more carefully. That's fine. But it turns out not to be quite that fine, because what we will find is you'll actually find a systematically exaggerated, systematically inflated link with, in this case, behavior, stronger than what it looks like you're finding. Essentially because it's not, this is called non-independence error. The problem is that the way I selected the region of interest, the ROI, is the very same, because then I go in there and I look for that very same thing that I just selected it on the basis of. It's like if I say, test me, I'm going to give some aptitude test to 1,000 people, and some people are going to score really highly. Well, some of those people are not really that great. They're just having a good day, or they got lucky when they guessed or something. So if you go in and you say, now I'm going to decide, evaluate my aptitude test by looking in and finding, did it really find people with good aptitude? Well, this group of people who you selected, they're all going to have pretty good aptitude because they all scored well on the test. So you have to be a little bit careful about figuring out, am I really finding out something about this group of people, or am I just finding out something about the way I actually selected them? So, so here's an example that they highlighted in the paper. And you can well imagine that the people who they kind of got highlighted in this way were not too happy about it. And maybe they actually had a right not to be too happy about it because, it wasn't the case that this was just a kind of egregious error. This was just kind of the way people did things. And some of these papers came out in Science and Nature. So this is one that Voodoo particularly highlighted. So this is actually a very interesting study, and I'm not certain, I haven't really followed this, but I think that people have done these kind of studies subsequently kind of doing the stats right and have still found similar things. So I don't think this means it was, a bogus result. I think maybe, they just got a slightly stronger result than it really looked at. So you say, find areas in the brain which correlate with the degree to which people feel pain when they're rejected, socially rejected. Okay. So the idea is that physical pain is really kind of like social pain. So they found this region in anterior cingulate, and they say, okay, and so this was selected on the basis of, correlating with behavior. So they say, okay, well, let's go take a closer look. Let's go look inside this area and look at the plot activation in this area against what's this social distress. Okay, you get a really, really, really strong correlation of 0.88. And those of you who do stuff with correlations will know you hardly ever get a correlation of 0.88. Okay? So the punchline is that there probably is a real correlation, but it's probably not nearly as strong as this looks. And the reason is because you've kind of biased yourself. You said, go into this area, find an area specifically because it has correlations, and then go in there and see just how good the correlation is. Well, yep, there's going to be a correlation there because that's how you found it in the first place. So there's something real going on, that's fine. But then the question is, how big is that thing that's going real? That's the part that gets inflated. Okay? So there's a nice little graph here. This is from the, this is not, I don't think this is in the paper that I said. I said the paper that's kind of a summary of the issue, but the original paper had this graph in it. And so this is the green and the red and the orange. They sent out this survey to people, and they said, how do you do your tests? And people, and so the people who kind of did the test right and actually were careful to kind of select the region in a way that was independent from the tests that they subsequently did in the region. And they found correlations. I don't know if you can see this here, but there's like correlations. Their correlations peaked around 0.5 or something, which is pretty decent correlation, but it's not crazy. Whereas people who did it in a non-independent way and who selected a region based on looking for a particular correlation and then went and tested for that same correlation in the same data, they got much higher correlations, going up to some people got correlations of almost 1, okay, which is kind of I don't know, I mean, 0.95, so not very many, but a lot of people got correlations, 0.8, 0.9, and usually, when you deal with noisy data like fMRI, this is kind of a rule of thumb. This is one reason why it wasn't obvious that they were messing up here, because sometimes you're really lucky, sometimes you get a really good correlation, but usually the world is messy. Usually, if you get a correlation like 0.85, 0.9, you've kind of messed up a bit. So what's the solution? The solution, remember how we talked about in using classifiers. People do this in classifiers all the time, and now people do it in kind of, at least in fMRI and regular stats too. Just divide up your data. So you have a training set, you take your data and you chop it. You don't have to chop it in 2, but you can chop it into 4 bits and rotate around which bit you use as a testing set, however you want to do it. But chop it up into different parts, and use one part as your training set. In this case, this is the part which you use for figuring out where's my ROI going to be. In this part of the data, in this training part of the data, find regions which have a good correlation with my behavioral measure. Then take the other part of the data and say, okay, now let's go and see how strong the correlation is in that part of the data, but using this ROI, the region of interest, that we define with the other part, and then you're okay. Okay, so it's actually so the good thing is it's actually a fairly simple solution, and it turns out to be exactly the same solution that people have been doing in machine learning for a long, long time, namely using cross-validation. So it's actually this story kind of has a happy ending, actually. Why is it good? Why is it good to divide things up into training and testing? Because it makes sure that you don't accidentally pollute yourself with your noise. It's exactly the same. It's exactly the same reason why people do it in machine learning. Sometimes noise very inconveniently disguises itself and looks just like signal. And when that happens, sorry, something weird just happened to my display. When that happens, when your noise looks just like signal, then it can easily lead you astray. And so the way to, I don't know why that happened, the way to stop that from happening is to divide your data into independent parts, and those independent parts are going to have independent noise in them too. So that's actually a fairly easy way to do it. And here it really is the case. People didn't really realize that this was an important thing to do, and now they do, and now people do this, and so you divide up the data. And so, however, here it's still actually not that uncommon to see papers which actually don't do this properly. Yeah.
So is there any problem other than like practicality with actually testing the correlation everywhere as opposed to seeing where are there heightened levels of activated boxes, so I'm going to look into that.
So you're saying why bother, why use a region of interest in the first place? Why not just look?
So is there any other reason other than we don't want to do it 30,000 times?
No, well that's a very, very good question. So the reason why people like to do region of interest analyses is because, especially if you kind of, this is not necessary, but especially if you subscribe to the view that there's kind of specialized regions in the brain that do particular things, it might be that the very question you're asking is, can I find, which part of the brain is, involved in perceiving social pain or something like that, right? So you actually want to find the region. That's kind of your question, and so it's not just the practicality of, well, I don't want to just do a test error, because that's actually your kind of research question. But also, looking more closely at doing a kind of big, broad search first, and then looking more closely at the stuff that kind of pops out is actually a very reasonable approach, right? I mean, that's like...
well, except if we don't separate it.
Yeah, exactly. So that's why, it wasn't nuts that people actually kind of stumbled into this problem without realizing it, okay? I mean, we do this all the time. You look, if you're trying to, I don't know, select... grad students to admit to a program, right? That's something we're kind of doing. Right? First you get loads and loads of applications, and then you make a kind of short list, and you say, oh, these guys look pretty good, and then you kind of like zoom in a bit on them, right? So this kind of thing happens all the time. And so that's essentially kind of what region of interest analysis too is saying, here's my kind of short list of, really interesting voxels. Let's go interview them.
So I guess my question is, would voodoo correlations arise if I just looked at all of the data that I had and put it next to the like signal model that I have, and I just concurrently analyzed every piece of data that I have.
Right. No, that's very good. No, this is a problem specifically from selection bias.
Okay.
Yeah. So it's a very, very good question. This only happens when you do selection.
Yeah.
And it's the bias, it's the selection that biases you.
Yeah.
And that you have to right. So it's a very, very good question. I mean, there's other types of problems that happen when, unfortunately, there's no way to escape, all problems. So if you don't do any selection, then you run more heavily into the multiple comparisons problem. So there's kind of different things to worry about at both ends. But anyway, these are problems that happen in fMRI. This whole discussion about Voodoo correlations, and there's this other paper. Where did I put that reference? Here you are. This previous quarter paper called it double dipping. This was only in 2009, so this is actually relatively recently. Even more recently, I'm running over, but even more recently, there's other types. There's always types of problems that people are uncovering, because human beings are just not good at thinking about this stuff. But ultimately, this is all a means to an end of finding stuff out there really in the world that really replicates. And so it's kind of like wearing a seatbelt. Okay, so that's the end of that. So on Thursday, we're going to be in the computer lab. Sorry, this is only registered students because there's not that much room. And we're going to do some more hands-on stuff of actually playing with real brain data. The same data actually that you kind of surfed around a bit last time, although this time the program should work on everyone's computers, I hope. And we're going to look at some of the pre-processing steps. And you're very, very welcome to bring your own laptop, but it won't be quite as easy as in the case, for instance, when you did, just those little MATLAB exercises, because this time there's actually like a whole bunch of data which you can download. I think I sent out this link, I can resend it, and also the SPM package, so you'll have to preload that stuff first if you want to do that. See you on Thursday.
Actually, seriously.
Will you? Are you sure?
Yes, sir.
Okay, because what's his face? Kelvin needs to be weighted clean. And I'm recording the. And I think that's it, which is good. Cool. I'll see you Thursday.
Hey, thank you. Very good.
I'm confused by the
Yeah, this is a tricky part of it. So I use something called the external question. Yeah, so there's a